So here you are, lying in the dark, and somewhere behind your closed eyes, inside the warm quiet of your skull, there's a universe running. Right now. Not metaphorically. I mean, there is an actual electrical storm happening between your ears. 100 billion tiny cells firing in patterns so complex that the most powerful supercomputers we've ever built can't fully simulate even a small fraction of what's going on in there. And the wild part, the part that might keep you up if you think about it too hard, is that the thing doing all that work is also the thing trying to understand itself. Your brain is the only object in the known universe that is actively trying to figure out what it is. That's either beautiful or deeply strange. And honestly, it's probably both. Let's just sit with that for a second. Every thought you've ever had, every memory of a summer afternoon or the smell of someone's cooking or the sting of an embarrassment you thought you'd forgotten, all of that lives in about 3 pounds of tissue that has the consistency, roughly, of firm gelatin. If you held a human brain in your hands, which neurosurgeons do regularly, you'd be surprised by how soft it is. It doesn't look like a supercomputer. It looks like a wrinkled, pinkish-gray walnut, the size of both your fists pressed together. And yet, it is, by any reasonable measure, the most complicated structure we have ever discovered. Not built. Discovered. Because nobody designed it. It assembled itself over hundreds of millions of years through the slow, blind, breathtakingly patient process of evolution. And understanding how it works, really understanding it, has been one of the longest, most humbling projects in the entire history of human knowledge. So tonight, let's walk through that story together. Not in a rush, not like there's a test tomorrow. Just slowly, the way you'd wander through a museum after hours when there's nobody else around and you can stop at whatever catches your eye. We'll start at the very beginning, and I mean the very beginning, thousands of years ago, when people first started wondering what the brain actually does. And we'll work our way forward through centuries of wrong guesses, brilliant accidents, heartbreaking failures, and moments of genuine revelation. Along the way, you'll start to see how this thing inside your head actually operates, piece by piece, system by system. And if you fall asleep somewhere in the middle, that's completely fine. That's actually your brain doing one of its most important jobs. We'll get to why sleep matters later. For now, just settle in. Here's something that might surprise you. For most of recorded history, people didn't think the brain was particularly important. Seriously. The ancient Egyptians, who were meticulous about preserving the body for the afterlife, they mummified everything, the heart, the liver, the lungs, the intestines, all carefully stored in canopic jars. But the brain? They pulled it out through the nose with a hook and threw it away. Just discarded it. Because they believed the heart was the seat of the soul, the center of thought and feeling and identity. The brain was just stuffing, packing material inside the skull. And honestly, if you think about it from their perspective, it kind of makes sense. When you're scared, you feel your heart pound. When you're in love, you feel a warmth in your chest. When you're nervous, your stomach churns. The brain doesn't announce itself like that. It just sits up there, quietly running everything, never once sending you a sensation that says, hey, I'm the one doing all this. It's the most important organ in your body, and it's also the most silent. The Greeks started to change that, slowly. Around the 5th century BCE, there was a real debate happening in the Mediterranean world about where the mind lived. On one side, you had people like Aristotle. Yes, that Aristotle, one of the most influential thinkers who ever lived. And he was firmly in the heart camp. He thought the brain was basically a cooling system for the blood, like a radiator. He noticed that the brain didn't seem to respond to touch the way other organs did, and that it was relatively cool compared to the heart, and he concluded that its job was to cool down the heat generated by the heart's passions. Which is wrong, obviously, but it's a beautiful example of how even brilliant people can build perfectly logical arguments on completely false foundations. But on the other side, there was Hippocrates. You've heard his name. He's the one behind the Hippocratic Oath that doctors still reference today. And Hippocrates, working around 400 BCE on the Greek island of Kos, made an observation that was genuinely ahead of its time. He wrote, and I'm paraphrasing here, that people ought to know that from the brain, and from the brain alone, arise our pleasures, joys, laughter, and sorrows, our pains, griefs, and tears. He was watching patients with head injuries, people who'd been struck in battle or fallen from heights, and he noticed something consistent. Damage to the head changed who people were. It changed how they spoke, how they moved, what they remembered, even their personalities. Damage to the heart killed you, sure, but damage to the brain could leave you alive and turn you into someone else entirely. That was the clue. That was the thread that, if you pulled on it long enough, would eventually unravel the whole mystery. But here's the thing. Nobody pulled on that thread for a very long time. Hippocrates was right, but being right and being believed are two very different things, especially in the ancient world. Aristotle's view, the heart-centered view, remained dominant for centuries, and even when people did start taking the brain seriously, they got distracted by the wrong part of it. Let me explain what I mean. If you were to cut a brain in half, and the ancient physicians did, mostly on animals, though a Roman physician named Galen in the 2nd century CE performed dissections on gladiators' wounds and occasionally on executed criminals, you'd see that the interior isn't solid. There are these hollow spaces inside, chambers filled with clear fluid. We call them ventricles. And for about 1,000 years, the prevailing theory in Western medicine was that these ventricles were where the action happened. The idea, which Galen helped develop and medieval scholars ran with, was that the fluid inside those chambers, they called it animal spirit or psychic pneuma, was the actual medium of thought. The solid brain tissue surrounding the ventricles? That was just the casing, the container. The real magic was in the liquid. It's called the ventricular doctrine, and it was remarkably detailed for being completely wrong. Medieval scholars mapped out a whole system. The front ventricle handled perception and common sense. The middle one handled reasoning and judgment, and the rear one stored memories. It was neat, it was tidy, and it dominated European thinking about the brain from roughly the 4th century all the way through the Renaissance. That's over 1,000 years of smart people looking at the brain and focusing on the wrong part of it. The ventricles, we now know, are important. They produce and circulate cerebrospinal fluid, which cushions the brain and clears waste. But they are not where thinking happens. Not even close. So what changed? Well, as with so many things in science, it started with someone being willing to actually look, really look, carefully, with fresh eyes and a sharp knife. In 1543, a young anatomist from Brussels named Andreas Vesalius published one of the most important books in the history of medicine. It was called De Humani Corporis Fabrica, on the fabric of the human body. And it was revolutionary, not because Vesalius had some grand new theory about the brain, but because he insisted on dissecting human cadavers himself with his own hands and drawing exactly what he saw. This might not sound radical, but it was. For centuries, anatomy lectures had been conducted with the professor sitting in a high chair reading aloud from Galen's ancient texts, while a barber surgeon did the actual cutting down below, and nobody checked whether what Galen wrote actually matched what was in front of them. Vesalius checked, and he found discrepancies everywhere. When it came to the brain, Vesalius produced illustrations of astonishing detail. The folds of the cortex, the structures deep inside, the ventricles in their true proportions. And here's what's subtle but important. By drawing the brain so carefully, Vesalius quietly shifted attention away from the ventricles and toward the brain tissue itself. He didn't necessarily have a better theory about how it all worked, but he gave future scientists something invaluable: an accurate map. He showed them what they were actually dealing with. Now, you might be wondering, if the ventricles aren't where thinking happens, then where does it happen? And the honest answer is that it took another 300 years after Vesalius before anyone really started to figure that out. 300 years. Think about what that means. Think about how patient science sometimes has to be. The brain was right there, inside every person who ever lived, and it took until the 1800s before we began to understand even the basics of how it operates. And the reason for that delay is actually fascinating, because it wasn't just a lack of curiosity or effort. It was a technology problem. The brain's secrets are hidden at a scale that the naked eye simply cannot reach. See, the real working units of the brain, the cells that do the actual computing, are tiny, unbelievably tiny. A single neuron, which is the type of cell we're talking about, has a cell body that's roughly 10 to 50 micrometers across. A micrometer is 1/1,000,000th of a meter. You could line up maybe 20,000 neurons side by side across your thumbnail. And the connections between them, the thread-like fibers that carry signals from one neuron to the next, can be thinner still. You simply cannot see this with your eyes. You can't see it with a magnifying glass. You need a microscope, and not just any microscope. You need a good one, with proper lenses and proper staining techniques to make the transparent cells visible against the transparent tissue surrounding them. And that technology didn't really come together until the late 19th century. Which brings us to one of the most remarkable chapters in the whole history of neuroscience, and it starts in a kitchen in northern Italy in the 1870s with a frustrated physician named Camillo Golgi. Golgi was working at a hospital for the chronically ill in Abbiategrasso, a small town near Milan. He didn't have a proper laboratory. He was doing his experiments at night, by candlelight, in his kitchen, and he was trying to solve a problem that had tormented anatomists for decades. When you looked at brain tissue under a microscope, everything was a blur. The cells were packed so tightly together, and they were so pale and translucent that you couldn't make out where one ended and another began. It was like trying to read a page where all the letters were printed in the same pale yellow ink on a white background. You knew something was there, but you couldn't resolve it. What Golgi did, almost by accident, was discover a staining method that changed everything. He was experimenting with silver nitrate, a chemical used in photography, and he found that if he soaked brain tissue in potassium dichromate and then exposed it to silver nitrate, something extraordinary happened. The silver compound would infiltrate individual neurons and turn them jet black against a pale yellow background. But here's the beautiful part, the part that makes this technique almost magical. For reasons that nobody fully understands even today, the stain only affects a tiny random fraction of the neurons in any given slice of tissue. maybe 1 or 2%. If it stained all of them, you'd be right back where you started, with an unreadable mess. But because it picks out just a few cells, scattered here and there, you can suddenly see each one in its entirety, the cell body, the long fibers stretching away from it, the delicate branching tree of smaller fibers reaching out in every direction. For the first time in history, a human being could look at a single brain cell and see its complete shape. Golgi published his findings in 1873, and the technique, called the Golgi stain, or sometimes the black reaction, eventually spread to laboratories across Europe. But it was another scientist, working in a very different country under very different circumstances, who would take Golgi's tool and use it to build the foundation of everything we now know about how the brain is organized. His name was Santiago Ramón y Cajal, and he was working in Spain, which at the time was considered a scientific backwater, far from the great research centers of Germany, France, and England. Cajal was the son of a rural physician in Aragon, and as a young man he had wanted to be an artist. His father disapproved, pushed him toward medicine, and Cajal eventually found a way to merge both passions, because what he did, starting in the late 1880s, was sit at his microscope for hours upon hours, studying Golgi-stained brain tissue and drawing what he saw with an artist's eye and a scientist's precision. And what he saw changed everything. What Cajal saw, peering through that microscope in his modest laboratory in Barcelona and later in Madrid, was a world of breathtaking intricacy. Each neuron revealed by the Golgi stain was its own little universe, a cell body, sometimes round, sometimes pyramidal, sometimes shaped like a star, with dozens or even hundreds of fine branches radiating outward like the limbs of a winter tree. And then, extending from one side of the cell body, a single long fiber, much thicker than the rest, stretching away into the distance, sometimes for what seemed like an enormous span relative to the size of the cell itself. Cajal drew these cells with extraordinary care, and his drawings, which still survive, and which you can find in archives and museums, are genuinely works of art. Delicate ink renderings, precise and elegant, capturing the branching patterns of individual neurons with a fidelity that even modern photography sometimes struggles to match. He wasn't just recording data. He was seeing something no one had ever properly seen, and he was determined to capture every detail. But here's where Cajal's contribution goes beyond beautiful pictures, because as he studied more and more tissue, from the brains of birds, mammals, human fetuses, adult humans, from the cerebellum, the cerebral cortex, the retina, the spinal cord, he began to notice something that contradicted the prevailing view of his time. The dominant theory, championed by many leading scientists, including Golgi himself, was called reticular theory. The idea was that the brain was essentially one giant continuous network, that all the fibers from all the neurons physically merged into each other, forming an unbroken web, like a fishing net where every strand connects to every other strand without any gaps. In this view, the brain wasn't made of separate cells at all. It was one enormous, fused mesh. And you can understand why people believe this. When you look at brain tissue, even stained brain tissue, the tangle of fibers is so dense and so complex that it really does look like everything is connected to everything else. The idea that there might be gaps, tiny spaces between one cell and the next, seemed almost counterintuitive. How could signals travel through the brain if the cells weren't physically touching? It didn't make sense, or at least, it didn't seem to make sense with the physics and chemistry people understood at the time. But Cajal, drawing after drawing, preparation after preparation, kept noticing something. The fibers from one neuron would approach the fibers of another neuron, get very close, sometimes agonizingly close, but they didn't fuse, they didn't merge. There was always a tiny gap. He could see it over and over in tissue after tissue, species after species. The neurons were individual, discrete cells. They came close to each other. They communicated with each other. That much was obvious from the fact that the brain clearly worked as a coordinated system, but they were separate entities. Each one was its own unit. This was the neuron doctrine, and Cajal spent decades building the case for it, publishing paper after paper, drawing hundreds upon hundreds of illustrations, arguing passionately at conferences and in journals. And the irony, the beautiful, strange irony of the whole thing, is that the tool he used to prove his case was Golgi's own stain. The very technique that Golgi had invented was now being used to demolish Golgi's own theory about how the brain was organized. And Golgi did not take this well. Not at all. The rivalry between Golgi and Cajal became one of the most famous feuds in the history of science. Golgi was a proud man, well-established, respected across Europe. Cajal was this upstart from Spain, working in relative obscurity, who had the audacity to use Golgi's own method to argue that Golgi was wrong about the most fundamental question in neuroscience. The tension between them simmered for years, and it came to a spectacular head in 1906, when the two men were jointly awarded the Nobel Prize in Physiology or Medicine. Think about that for a second. They shared the Nobel Prize. They both had to travel to Stockholm. Both had to give acceptance speeches, and Golgi, rather than graciously acknowledging the state of the science, used his Nobel lecture to attack Cajal's neuron doctrine and defend reticular theory. Right there, on the biggest stage in science, Golgi essentially argued that the man sitting next to him was wrong about everything important. Cajal, by all accounts, was furious but kept his composure, and history has thoroughly vindicated him. The neuron doctrine is now one of the foundational principles of neuroscience. Your brain is made of individual cells, roughly 86 billion of them, give or take, and they communicate across tiny gaps. Now, those tiny gaps. Let's talk about those for a second, because they turn out to be one of the most important features of the entire system. The gap between one neuron and the next is called a synapse, a term coined by the English physiologist Charles Sherrington around 1897. Sherrington didn't actually see synapses. They're far too small to see with the light microscopes of that era, only about 20 to 40 nanometers across, which is thousands of times thinner than a human hair. He inferred their existence from his experiments on reflexes in animals. He was studying how signals traveled through the spinal cord, and he noticed that reflexes had certain properties that didn't make sense if the nervous system was one continuous wire. There were tiny delays. Signals could travel in one direction but not the other. Responses could be strengthened or weakened depending on what other signals were happening at the same time. All of these observations pointed to the existence of some kind of junction point, some place where the signal had to cross from one cell to the next, and where something interesting could happen during that crossing. And this is where it gets really fascinating, because the synapse isn't just a gap that signals have to jump across. It's a decision point. It's a place where information gets processed. Think of it this way. If neurons were just wires soldered together end to end, the brain would be nothing more than a very complicated circuit, and a signal that started at one end would just travel to the other end, unchanged. But because there are synapses, because there are these tiny gaps, where the signal has to be actively transmitted from one cell to the next, the brain gains the ability to modulate, to adjust, to compute. A synapse can make a signal stronger or weaker. It can let a signal through or block it. It can combine signals from multiple sources and produce a different output depending on the combination. This is where the magic happens. This is where thinking lives. But here's the thing. Figuring out how signals actually cross that gap took another 50 years of painstaking work after Sherrington named it. And the story of how that happened involves one of the most elegant experiments in the history of biology, performed by a man who got the idea from a dream. Otto Loewy was an Austrian-born pharmacologist who, by the 1920s, had been puzzling over a question that was dividing the scientific community. When a nerve signal reaches the end of a neuron and arrives at a synapse, how does it get across to the next cell? There were two competing camps. One group believed it was electrical, that the signal simply jumped across the gap as a tiny spark of electricity, the way a spark jumps across the gap in a spark plug. The other group believed it was chemical, that the arriving signal caused the first neuron to release some kind of chemical substance into the gap, and that chemical substance then triggered the next neuron to fire. The electrical camp had simplicity on their side. The chemical camp had some suggestive evidence, but no proof. Loewy later told the story, and it's one of those stories that's almost too good to be true, but he told it consistently throughout his life. That on the night before Easter Sunday in 1921, he woke up in the middle of the night with the design for a perfect experiment fully formed in his mind. He scribbled some notes on a scrap of paper, went back to sleep, and in the morning found that he couldn't read his own handwriting. He spent the entire day trying to remember what the idea had been, and couldn't. But then, the following night, the idea came back to him in another dream, and this time, he got up immediately, went to his laboratory, and performed the experiment before dawn. What he did was beautifully simple. He took 2 frog hearts. Frogs were a standard experimental animal for this kind of work because their hearts keep beating for a long time, even when removed from the body. He kept both hearts alive in a bath of saline solution. One heart still had its vagus nerve attached. That's the nerve that, when stimulated, slows the heart rate down. He stimulated the vagus nerve of the first heart, and sure enough, it slowed down. Then he took the saline solution that had been bathing that first heart and transferred it to the second heart, the one with no nerve attached at all, and the second heart slowed down too. Think about what that means. There was no electrical connection between the 2 hearts. No wire, no nerve, nothing. The only thing that passed between them was the liquid, which meant that when the vagus nerve fired, it must have released some chemical substance into the fluid, and that chemical substance was enough to affect the second heart, all by itself. The signal wasn't electrical, it was chemical. Loewy called the mystery substance Vagusstoff, vagus substance, and it was later identified as acetylcholine, which turned out to be one of the first neurotransmitters ever discovered. And that word, neurotransmitter, is one you've almost certainly heard before, even if you've never studied neuroscience. Serotonin, dopamine, norepinephrine, GABA, glutamate. These are all neurotransmitters, chemical messengers that carry signals across synapses. When people talk about depression being related to serotonin levels, or Parkinson's disease being caused by a loss of dopamine-producing neurons, or anxiety medications working on GABA receptors, they're talking about this exact system, the chemical signaling system that Loewy first proved existed with 2 frog hearts and a dish of salt water on an Easter morning in 1921. He won the Nobel Prize for it in 1936, and his discovery opened up an entire universe of research that continues to this day. But let's slow down for a moment and think about what we've established so far, because we've actually covered an enormous amount of ground. We started with the ancient world, where people weren't even sure the brain mattered. We moved through centuries of wrong guesses and slow progress, through the ventricular doctrine, through Vesalius redirecting attention to brain tissue itself, through the long technological drought where nobody could see what neurons actually looked like. Then, Golgi's stain cracked open the door, Cajal walked through it, and established that the brain is made of billions of individual cells. Sherrington inferred that those cells communicate across tiny gaps called synapses, and Lowe proved that the communication is chemical. Each of these steps took decades. Each one was fought over, doubted, resisted, and each one was absolutely necessary before the next one could happen. Now, here's what's interesting about where we are in the story. By the middle of the 20th century, scientists understood the basic architecture: neurons, synapses, chemical transmission. But they still didn't really understand how a single neuron actually works. How does a neuron decide to fire? What happens inside the cell when it sends a signal? How does an electrical impulse travel down that long fiber, the one Cajal drew so carefully, from the cell body all the way to the synapse at the far end? These questions might sound simple, but answering them required a completely different kind of science, one that involved electricity, ion channels, and a very unusual animal that nobody would have expected to find in a neuroscience laboratory: a squid. Specifically, the Atlantic longfin squid, Loligopiale, a creature about a foot long that darts through the coastal waters off places like Cape Cod and Woods Hole, Massachusetts. And the reason this humble squid changed everything about our understanding of the brain comes down to one remarkable anatomical feature. It has a giant axon. Not giant like slightly bigger than usual. Giant like you can actually see it with your naked eye. Most axons in the human brain are incredibly thin, maybe a micrometer or two across. You'd need a powerful microscope just to find one. But the squid's giant axon, which controls the animal's jet propulsion escape response, can be up to 1 millimeter in diameter. That's roughly 1,000 times wider than a typical human nerve fiber. And that matters enormously because in the 1930s and 1940s, when scientists were desperate to understand how electrical signals travel along a nerve, they needed something big enough to stick an electrode inside. This is where two young British physiologists enter the story, and their names are Andrew Huxley and Alan Hodgkin. They were both at Cambridge, both brilliant, both fascinated by the same fundamental question: how does a nerve impulse actually work? People had known since the late 1700s, since Galvani's famous experiments with frog legs, that nerves had something to do with electricity. But the details were completely mysterious. How does a nerve cell generate an electrical signal? How does that signal propagate along the length of the axon without fading out? And how does the signal know to go in one direction? Hodgkin and Huxley began their collaboration in 1939, and their timing was spectacularly unlucky. They managed to get some beautiful early recordings from squid giant axons at the Marine Biological Laboratory in Plymouth, England, and then World War II broke out. Hodgkin spent the war years working on radar. Huxley worked on gunnery problems. 6 years of their scientific prime interrupted. But here's the thing. Those wartime detours weren't entirely wasted. Working on radar and military electronics gave both of them deep experience with electrical circuits, amplifiers, feedback systems. And when they came back to the squid axon after the war, they brought with them not just sharper technical skills, but a whole way of thinking about electrical systems that turned out to be exactly what they needed. So let me explain what they found, because it's one of the most elegant pieces of science in the 20th century. And it explains something happening inside your head right now, billions of times per second as you listen to this. Picture a neuron. You've got the cell body, which is where the nucleus lives, and extending out from it is this long, thin fiber, the axon. The axon is basically a cable, and its job is to carry electrical signals from one end to the other. Now that axon is surrounded by a membrane, a thin, fatty layer that separates the inside of the cell from the outside. And here's what's crucial. The fluid inside the cell and the fluid outside the cell have different concentrations of charged particles, ions. Specifically, there's a lot more potassium inside the cell and a lot more sodium outside. This imbalance creates a voltage difference across the membrane, like a tiny battery. When the neuron is just sitting there, not firing, this voltage difference is about -70 millivolts. That's the resting membrane potential. It's not much, 70 thousandths of a volt, but it's everything. Now, embedded in that membrane are proteins that act as tiny gates, ion channels. Some of them let sodium through, some let potassium through, and critically, many of them are voltage gated, meaning they open and close depending on the voltage across the membrane. This is where it gets really beautiful. When a neuron receives enough stimulation, say, from neurotransmitters hitting its dendrites, the voltage at a particular spot on the membrane starts to rise, becoming less negative. If it rises enough to cross a threshold, around -55 millivolts, something dramatic happens. The voltage-gated sodium channels at that spot snap open. Sodium ions, which have been concentrated outside the cell, come rushing in. They're positively charged, so they make the inside of the cell suddenly positive. The voltage shoots up from -70 all the way to about +30 or 40 millivolts in less than a millisecond. This rapid spike is the action potential. That's the signal. That's the fundamental unit of information in your nervous system. But it doesn't stop there. Almost immediately after those sodium channels open, they automatically inactivate. They slam shut on their own, like a gate with a built-in timer. And at the same time, voltage-gated potassium channels open. Potassium rushes out of the cell, carrying positive charge with it, and the voltage plummets back down. In fact, it briefly dips even lower than the resting potential. That's called the refractory period, and it's important because it means the neuron can't fire again right away. It needs a moment to reset. Think about that for a second. The whole thing, the rise, the spike, the fall, the reset, takes about 2 milliseconds. It's astonishingly fast. And here's what's really clever about how this propagates down the axon. When sodium rushes in at one spot and makes it positive, that positive charge spreads a little way along the inside of the axon, like ripples in a pond. When it reaches the neighboring patch of membrane, it pushes that patch above threshold 2, and those sodium channels pop open. Then the next patch, then the next. The action potential doesn't just sit in one place, it cascades down the length of the axon like a wave. And because each patch enters that refractory period right after firing, the wave can only move forward. It can't turn around and go backward. So you get a one-directional self-regenerating electrical signal that travels the full length of the axon without losing strength. It's not like an electrical current in a wire, which gets weaker with distance. Every patch of membrane regenerates the signal fresh. It's more like a line of dominoes, where each one falls with the same force, no matter how far down the chain you are. What Hodgkin and Huxley did between 1947 and 1952 was figure all of this out in extraordinary mathematical detail. They used a technique called the voltage clamp, which let them artificially hold the membrane at a specific voltage and measure exactly how much current flowed through it, and which ions were carrying that current. It was painstaking work. They'd thread a thin silver wire down the inside of a squid giant axon, set the voltage, and measure the currents. Then they'd change the solution bathing the axon, remove the sodium, for instance, and see what happened to the currents. By systematically varying conditions, they teased apart the separate contributions of sodium and potassium channels. And then, and this is the part that still amazes people, they wrote a set of differential equations that described the whole process. The Hodgkin-Huxley model, published in 1952, predicted the shape of the action potential, its speed, its threshold, even the refractory period, all from first principles. They didn't have computers in any modern sense, so Huxley ground through the calculations on a hand-cranked mechanical calculator. It took him weeks of tedious arithmetic to compute what a laptop could do in a fraction of a second today. The results matched their experimental data almost perfectly. They won the Nobel Prize in 1963, and their model remains the foundation of computational neuroscience to this day. Every simulation of neural activity, every artificial neural network that tries to mimic biological neurons at a detailed level, starts from the framework Hodgkin and Huxley built using squid axons and a hand-cranked calculator in post-war Cambridge. Now, there's a natural question you might be asking. If the action potential travels down the axon like a wave, patch by patch, how fast does it go? And the answer is, it depends. In a bare, unmyelinated axon, the speed is roughly proportional to the diameter of the fiber. Bigger axon, faster signal. That's exactly why the squid evolved that giant axon in the first place. It needs a fast escape response, and making the axon enormous was evolution's brute force solution. But vertebrates, including you, came up with a much more elegant trick, and it's called myelination. Myelin is a fatty, insulating substance produced by specialized glial cells, oligodendrocytes in the brain and spinal cord, Schwann cells in the peripheral nerves. These cells wrap themselves around the axon in tight spirals, like electrical tape wound around a wire, forming a thick insulating sheath. But they don't cover the whole axon. Every millimeter or so, there's a tiny gap where the axon membrane is exposed. These gaps are called nodes of Ranvier, named after the French anatomist Louis-Antoine Ranvier, who described them in the 1870s. And here's the beautiful part. The action potential doesn't have to slowly cascade through every patch of membrane anymore. Instead, it jumps from one node to the next. The insulation prevents current from leaking out through the myelinated segments, so the electrical signal shoots quickly through those covered stretches and then regenerates only at the nodes. This jumping conduction is called saltatory conduction, from the Latin saltare, meaning to leap, and it's dramatically faster. A myelinated axon in your body can conduct signals at up to 120 meters per second. That's over 260 miles per hour. An unmyelinated fiber of the same diameter might manage less than 1 meter per second. So myelination gives you a 100-fold speed increase without needing the axon to be enormous. It's a brilliant engineering solution, and it's why your brain can fit billions of fast-conducting nerve fibers into a space the size of a cantaloupe, while the squid needs a single axon, the width of a pencil lead, just to manage one escape reflex. And this isn't just an abstract anatomical fact. You've actually heard of what happens when myelin breaks down, even if you didn't know that's what was happening. Multiple sclerosis, MS, is a disease where the immune system attacks myelin in the brain and spinal cord. When those insulating sheaths are damaged, signals slow down, become erratic, or fail entirely. The symptoms depend on where the damage occurs: vision problems, muscle weakness, numbness, difficulty with coordination. It's a devastating illustration of just how critical that fatty wrapping is to normal brain function. The neurons are still there, the synapses are still there, but without intact myelin, the communication system falls apart. So now, we have a much richer picture than we had even a few minutes ago. We've got neurons as individual cells, separated by synaptic gaps. We've got chemical neurotransmitters carrying signals across those gaps. We've got the action potential, this elegant, self-regenerating electrical wave carrying signals along the axon. And we've got myelin speeding the whole thing up to remarkable velocities. But there's a piece of this puzzle we haven't really examined yet, and it might be the most important one of all. Because so far, we've mostly talked about single neurons doing their thing. One cell fires, the signal travels down its axon, neurotransmitter crosses a synapse, the next cell responds. That's the basic unit. But your brain contains something like 86 billion neurons, and each one can form thousands of synaptic connections. Some neurons receive input from 10,000 other neurons simultaneously. The total number of synapses in a human brain is estimated at somewhere around 100 trillion. And the real magic of the brain, the thing that makes it a brain and not just a bundle of wires, emerges from the patterns of those connections. How neurons are wired together, how those connections strengthen or weaken over time, how networks of thousands or millions of cells coordinate their activity to produce something as complex as a memory or a decision or the feeling of being in love or recognizing your mother's face in a crowd. That's what we need to talk about now. Because understanding a single neuron is like understanding a single musician. You can study how they hold the instrument, how they read the notes, how their fingers move. But that doesn't tell you what a symphony sounds like. And the person who first articulated this idea in a way that changed everything was a Canadian psychologist named Donald Hebb. Hebb published a book in 1949 called The Organization of Behavior, and in it, he proposed something that sounds almost too simple to be revolutionary. His idea, paraphrased in the way neuroscientists love to quote it, goes like this: neurons that fire together, wire together. That's the catchy version. What Hebb actually wrote was more careful. He suggested that when one neuron repeatedly participates in firing another neuron, when cell A consistently helps activate cell B, then something changes at the connection between them. That synapse gets stronger. The connection becomes more efficient, more likely to succeed the next time. And if they stop firing together, the connection weakens. Think about what that means. It means the brain isn't a fixed circuit. It's not like the wiring in your house, where the connections are soldered in place and that's that. The brain is a system that rewires itself based on experience. Every time you learn something, every time you practice a skill, every time something happens to you that matters, the pattern of synaptic strengths in your brain shifts. Some connections get turned up, others get turned down. The network reconfigures. And Hebb proposed this in 1949, before anyone had the tools to actually prove it at the cellular level. He was reasoning from psychology, from behavior, from what he observed about how animals and humans learn. It was a theoretical leap, and it was remarkably prescient. Now, Hebb's life is interesting in its own right. He grew up in Nova Scotia, struggled as a student early on, actually worked as a farmer and a laborer before going back to school. He ended up studying under some of the biggest names in psychology and neuroscience of his era: Carl Lashley at Harvard, Wilder Penfield at the Montreal Neurological Institute. Penfield, by the way, was doing extraordinary work at the time, electrically stimulating the brains of conscious patients during surgery and mapping out which regions controlled which functions. Patients would report vivid memories, sensations, movements, all triggered by a tiny electrode touching their exposed cortex. Hebb absorbed all of this. He saw the clinical data. He thought deeply about how networks of neurons could give rise to complex behavior, and he synthesized it into his theory. But here's the thing: Hebb's rule was a prediction, a beautiful, elegant prediction, and it took about another quarter century before someone actually found the biological mechanism that made it real. That someone was Tim Bliss, working with Terje Lomo at a lab in Oslo, Norway. In 1973, they published a paper describing something they'd observed in the hippocampus of rabbits. The hippocampus, and we'll come back to why this particular brain region matters so much, is a curved structure tucked deep inside the temporal lobe, and it had already been implicated in memory formation. What Bliss and Lomo found was this: if you stimulated a pathway in the hippocampus with a rapid burst of high-frequency electrical pulses, the synapses along that pathway became stronger, not for a few seconds, not for a few minutes, for hours, days, in some experiments, weeks. They called it long-term potentiation, LTP for short, and it was exactly the kind of mechanism Hebb had imagined. Repeated, intense co-activation of neurons led to a lasting strengthening of the connection between them. LTP turned out to be one of the most studied phenomena in all of neuroscience. Thousands of papers have been written about it, and as researchers dug into the molecular details, they found a whole cascade of events underlying it. One of the key players is a receptor called the NMDA receptor. That stands for N-methyl-D aspartate, which is just the name of the chemical used to identify it. Don't worry about the name. What matters is what it does. The NMDA receptor sits on the postsynaptic neuron, the receiving cell, and it has this incredibly clever property. It's what scientists call a coincidence detector. It only opens fully when two things happen at the same time. The presynaptic neuron has to release glutamate, which is the brain's main excitatory neurotransmitter, and the postsynaptic neuron has to already be somewhat depolarized, already somewhat active. Both conditions simultaneously. If only one happens, the channel stays mostly blocked by a magnesium ion sitting right in its pore. But when both happen together, when the sending cell and the receiving cell are active at the same time, that magnesium ion gets knocked out of the way, the channel opens, calcium floods in, and that calcium triggers a whole chain of molecular events that ultimately strengthen the synapse. It's Hebb's rule, implemented in biochemistry. Neurons that fire together wire together, and the NMDA receptor is the molecular gatekeeper that detects the coincidence. Now, the strengthening itself takes different forms depending on the timescale. In the short term, the first minutes to hours, existing proteins at the synapse get modified. Receptors get shuffled around in the membrane. More of them get inserted into the postsynaptic surface, making it more sensitive to future signals. But for truly long-lasting changes, the kind that underlie memories you keep for years, something more dramatic has to happen. The signal has to reach the nucleus of the cell. Gene expression has to change. New proteins have to be manufactured and shipped out to that specific synapse to physically remodel it, make it bigger, more robust. There's even evidence that entirely new synaptic connections can sprout. The architecture of the brain literally changes shape when you learn something that sticks. And here's what's really beautiful about this. It goes both ways. Just as synapses can be strengthened, they can also be weakened. There's a complementary process called long-term depression, LTD, which is a terrible name because it has nothing to do with mood. It just means a long-lasting decrease in synaptic strength. Low frequency, poorly timed stimulation can cause synapses to become less effective. Receptors get pulled out of the membrane. The connection gets quieter. This is just as important as strengthening, because if all your synapses only ever got stronger, the whole system would eventually saturate. You'd lose the ability to distinguish signal from noise. The brain needs to be able to both turn connections up and turn them down. It's the balance between LTP and LTD that allows the network to stay flexible, to keep learning new things without losing the ability to function. So let's pause here and appreciate the picture that's emerging. You've got 86 billion neurons connected by 100 trillion synapses, and every one of those synapses has a strength that can be adjusted up or down based on the pattern of activity flowing through the network. The whole system is in constant flux, constantly being sculpted by experience. This is what neuroscientists mean when they talk about plasticity, the brain's ability to change itself. And it's not just something that happens during childhood, though it's certainly most dramatic then. Adult brains are plastic too. Every time you learn a new route to work, every time you pick up a new word in a foreign language, every time you get better at a video game or adjust to a new pair of glasses, synaptic strengths are shifting somewhere in your brain. But plasticity needs to be understood in context because the brain isn't just a uniform soup of neurons all connected to each other randomly. It has structure, tremendous structure. And that brings us to something we should talk about, how the brain is organized on a larger scale and how we figured that out. The story really begins with a series of unfortunate injuries, because for most of history, the main way humans learned about what specific brain regions do was by observing what happened when those regions were damaged. And no case in the history of neuroscience is more famous than that of Phineas Gage. It was September 13th, 1848, near Cavendish, Vermont. Gage was a 25-year-old railroad construction foreman, by all accounts a capable, well-liked, responsible man. His crew was blasting rock to clear a path for new rail lines, and the process involved drilling holes into rock, filling them with gunpowder, laying a fuse, and then using a large iron rod, a tamping iron, to pack sand over the charge. Gage's tamping iron was custom made. It was 3 feet 7 inches long, weighed over 13 pounds, and tapered to a point about 1/4 inch in diameter. On that September afternoon, something went wrong. The powder ignited prematurely. The tamping iron was launched like a rocket straight through Gage's skull, entering below his left cheekbone, passing behind his left eye, through the front of his brain, and out through the top of his head. It landed some 80 feet away, smeared with blood and brain matter. And Phineas Gage survived. He was conscious within minutes. He reportedly spoke to the men around him. He was taken by ox cart to his lodgings, where a local physician named John Martin Harlow treated him. The physical recovery, while complicated by infection, was remarkable. Within a couple of months, Gage was up and about, physically functional, but he was different. And this is where the case becomes so important. The people who knew Gage before the accident said he was no longer himself. Dr. Harlow's later reports described a man who had become fitful, irreverent, profane, impatient, unable to stick with plans, dismissive of advice. The railroad company, which had regarded him so highly before, wouldn't take him back. His friends said he was, in Harlow's famous phrase, no longer Gage. Now, we should be careful here, because the Gage story has been somewhat mythologized over the years. Some of the more dramatic claims about his personality change are based on limited and possibly embellished accounts. Modern researchers like Malcolm McMillan have done painstaking work trying to separate fact from legend. What does seem clear is that there was a meaningful change in Gage's behavior and personality, and that the damage was concentrated in his prefrontal cortex, the front part of the frontal lobes. And that was the revelation. Here was a man whose ability to move, to speak, to see, to remember, all of that was largely intact. But something about his judgment, his social behavior, his ability to plan and regulate his impulses was profoundly altered. It suggested that the frontal lobes weren't just filler or some kind of reserve tissue. They were doing something specific and crucial, something related to personality, decision-making, social conduct, the very qualities that make you you. Gage's case wasn't the only evidence pointing in this direction, but it became the most iconic. And it opened the door to a much bigger question that consumed neuroscience for the next century and a half. To what extent is the brain a collection of specialized regions, each handling a specific function, versus a more holistic organ where everything contributes to everything? This debate, localization versus holism, has a long and surprisingly contentious history, and it shaped the careers of some of the most brilliant and stubborn scientists who ever studied the brain. On one side, you had people like Paul Broca, a French physician who in 1861 examined a patient known as Tan. That was the only syllable the man could produce, despite being able to understand language perfectly well. When Tan died and Broca examined his brain, he found a lesion in the left frontal lobe, in a region now called Broca's area. A few years later, a German neurologist named Karl Wernicke identified a different region, further back in the left hemisphere, where damage caused a different kind of language problem. Patients could speak fluently, but what they said made no sense, and they couldn't understand what others said to them. Wernicke's area, as it came to be known, was clearly doing something different from Broca's area, even though both were involved in language. These discoveries were powerful evidence for localization. Specific brain regions seemed to handle specific functions. But the picture was never as clean as the textbook diagrams suggest, and there were always researchers who pushed back, who argued that the brain worked more as an integrated whole. Carl Lashley, the same Lashley who mentored Donald Hebb, spent decades in the early 20th century trying to find where memories were stored in rat brains. He trained rats on mazes, then systematically destroyed different parts of their cortex, expecting to find the spot where the memory lived. He never found it. No matter which piece he removed, the rats could still partially remember the maze. What mattered more was how much cortex was destroyed, not which specific piece. He called this the principle of mass action, and it led him to his famous, slightly exasperated conclusion that memory seemed to be everywhere and nowhere at once. Lashley was wrong in some ways. We now know that different types of memory do depend on different brain structures, quite specifically. But he was also onto something real, because the brain does distribute many functions across networks, rather than pinning them to single locations. The truth, as it usually does in neuroscience, turned out to be more nuanced than either camp imagined. and the case that did more than perhaps any other to reveal how memory actually works in the brain, the case that bridged the gap between Lashley's frustration and our modern understanding, involved a man who, for decades, was known to the world only by his initials, HM. His real name, revealed after his death in 2008, was Henry Myelination. And what happened to Henry changed everything we thought we knew about how the brain stores the past. Henry Myelination was born in 1926 in Manchester, Connecticut. As a child, he began suffering from severe epileptic seizures, possibly triggered by a bicycle accident when he was 7, though that connection is debated. By the time he was in his late 20s, the seizures had become so frequent and so debilitating that he could barely hold a job or live a normal life. Medications weren't controlling them. He was desperate. His family was desperate. And in 1953, a neurosurgeon named William Beecher Scoville offered a radical option. Scoville proposed removing the medial portions of both temporal lobes, the inner parts of the brain on each side, tucked beneath the temples, because that's where the electrical storms of Henry's seizures seemed to originate. The surgery involved removing, bilaterally, structures including most of the hippocampus on both sides, along with the amygdala and surrounding cortex. It was an aggressive procedure, and at the time, the full consequences of removing both hippocampi were not well understood. The surgery worked, in a sense. Henry's seizures became much more manageable, but something else happened, something no one had anticipated with such devastating clarity. Henry could no longer form new long-term memories. He could remember his childhood. He could recall events from years before the surgery, though some of the years just preceding it were hazy. He could hold a conversation, follow what you were saying, respond intelligently. His IQ was actually measured as slightly above average after the operation. His personality was intact. People described him as gentle, cooperative, good-humored. But the moment you left the room and came back 5 minutes later, he had no idea who you were. He could read the same magazine over and over and find it fresh each time. He couldn't remember what he'd eaten for breakfast. He lived from 1953 until his death in 2008 in a kind of permanent present tense, aware, conscious, able to think and speak, but unable to carry new experiences forward into lasting memory. Think about what that means for a moment. 55 years of life after the surgery, and almost none of it stuck. Every day was, in some sense, his first day. The people who studied him for decades had to reintroduce themselves every single visit. He knew something was wrong. He'd sometimes say he felt like he was waking from a dream. But he couldn't hold on to even that realization long enough to fully grapple with it. The person who studied Henry most extensively was Brenda Milner, a British-Canadian neuropsychologist working at the Montreal Neurological Institute. Milner had already been investigating memory and the temporal lobes when Scoville contacted her about Henry's case. She began testing him in 1955, and what she found over the following years reshaped the entire field. The first and most obvious conclusion was that the hippocampus, or at least the medial temporal lobe structures that had been removed, was essential for converting short-term experiences into long-term memories. This process would later be called consolidation. Henry could hold information in mind for seconds, maybe a minute or two, as long as he was actively thinking about it. That's short-term or working memory, and it was intact. But the bridge from short-term to long-term was destroyed. The hippocampus, it turned out, was that bridge. But here's where it gets really interesting, and this is the part that made Henry's case not just tragic, but scientifically revolutionary. Milner discovered that Henry could still learn certain things. In one famous experiment, she asked him to trace a star shape while looking only at his hand's reflection in a mirror. A tricky task, because the mirror reverses everything, so your hand moves the opposite direction from what you expect. It's frustrating at first. You make lots of errors. But with practice, you get better. And Henry got better. Over 3 days of testing, his performance improved steadily, just like anyone else's would. The thing is, each day, he had no memory of ever having done the task before. He'd sit down, Milner would explain it, and he'd say something like, "Well, this is new." But his hands knew. His motor system had learned, even though his conscious mind had no record of the learning. This was the critical insight. There isn't just one kind of memory. There are multiple memory systems in the brain, and they depend on different structures. The kind of memory Henry lost, the ability to remember events, facts, experiences, the story of your own life, that's called declarative memory, and it depends heavily on the hippocampus and surrounding medial temporal lobe. But the kind of memory that let him improve at mirror tracing, procedural memory, the memory for skills and habits, that relies on different brain circuits, particularly the basal ganglia and the cerebellum, structures that were left intact by Scoville's surgery. Henry's case proved that these systems could be separated. You could lose one and keep the other. And the distinctions kept multiplying as researchers studied him and other patients with similar injuries. Declarative memory itself turned out to have subtypes. There's episodic memory, your personal autobiography, the memory of specific events you experienced, like what you did last Tuesday or the first time you rode a bicycle. And there's semantic memory, your storehouse of general knowledge and facts, like knowing that Paris is the capital of France, or that water freezes at 0 degrees Celsius. These are related but not identical. Some patients with hippocampal damage lose episodic memory more severely than semantic memory, suggesting the two systems, while overlapping, aren't completely the same circuit. Then there's the whole category of non-declarative or implicit memory, the things you learn without being consciously aware you've learned them. Procedural memory is one type, but there are others. There's priming, where exposure to a stimulus makes you faster or more likely to respond to related stimuli later, even if you don't remember the original exposure. There's classical conditioning, the kind Pavlov demonstrated with his dogs, and even that has subtypes, some of which depend on the cerebellum, and some on the amygdala, depending on whether the response involves skeletal muscles or emotional reactions. Henry could be conditioned to show certain emotional responses to stimuli, even though he couldn't remember the conditioning sessions. His amygdala, or what remained of it, could still learn fear associations, even when his hippocampus couldn't file the experience as a conscious memory. So what emerged from Henry's case, and from the decades of research it inspired, was a taxonomy of memory, a map showing that the brain doesn't have one filing cabinet, but several, each handling different kinds of information, each relying on different neural hardware. The hippocampus is crucial for declarative memory, especially for forming new episodic memories. The basal ganglia handle habits and skills. The cerebellum is essential for motor learning and certain conditioned responses. The amygdala processes emotional memory, particularly fear. And the neocortex, the vast, folded outer surface of the brain, seems to be where long-term declarative memories eventually settle once the hippocampus has done its initial consolidation work. That last part is worth lingering on because it circles back to Lashley's old puzzle. Remember how Lashley couldn't find the memory in any one spot in the cortex? He was looking in the right place in a way. Long-term memories are stored across the cortex, but he was looking at the wrong time. A fresh memory depends on the hippocampus. Over days, weeks, months, and even years, that memory gradually becomes independent of the hippocampus and is stabilized in cortical networks. This process, called systems consolidation, is thought to happen partly during sleep, which is a nice irony given what you're doing right now. While you sleep, the hippocampus replays recent experiences, strengthening the cortical connections that will eventually hold those memories on their own. It's as if the hippocampus is a teacher who drills the cortex on new material every night until the cortex knows it well enough to go it alone. This is why Henry could remember his childhood, but not what happened yesterday. His old memories, formed before the surgery, had already been consolidated into cortical networks and no longer needed the hippocampus. But new experiences never got that chance. They entered his awareness, lingered briefly in working memory, and then simply faded, like words written on water. Henry Myelination died on December 2nd, 2008, at the age of 82. After his death, his brain was donated to science, and it was sliced into over 2,000 thin sections by a team led by Jacopo Annese at the University of California, San Diego, in a marathon 53-hour session that was streamed live online. The detailed anatomical analysis confirmed exactly which structures Scoville had removed and allowed researchers to correlate the precise extent of the damage with the specific memory deficits Henry had shown over his lifetime. His contribution to neuroscience is almost impossible to overstate. More than 100 scientific papers were written about him. He is arguably the most studied single patient in the history of brain science. And yet, here's something that should give you pause. Henry never knew any of this. He couldn't remember the tests, the researchers, the decades of visits. He lived his life pleasantly by most accounts. He was described as unfailingly polite, often cheerful, but he existed in a kind of timeless bubble, unable to build the narrative thread that most of us take for granted as the substance of a life. His case raises questions that go beyond neuroscience into philosophy. What does it mean to be a person if you can't remember your own past? Is identity something that requires continuity of memory? Henry seemed like himself to people who met him, warm, present, engaged. And yet he couldn't tell you who he'd been 5 minutes ago. But those are questions for another kind of late-night conversation. For now, what matters is what Henry's case taught us about the architecture of memory, and how that understanding opened the door to studying something even more complex, the question of how the brain creates not just memories, but awareness itself. Because once you've mapped the memory systems and understood that different circuits handle different functions, you start to wonder about the thing that ties it all together. You start to wonder about consciousness. And that's a question that has haunted neuroscience from the very beginning. A question that some of the most brilliant minds of the 20th century tried to crack open, and that many of them eventually admitted might be the hardest problem in all of science. Not just hard the way quantum mechanics is hard, where the math is brutal, but the experiments eventually give you answers. Hard in a different way. Hard because consciousness seems to resist the very tools we've built to study it. Think about what we're really asking here. We've talked about neurons firing, about action potentials racing down axons, about neurotransmitters crossing synaptic gaps, about circuits strengthening through long-term potentiation. All of that is mechanism. It's the machinery doing things, processing signals, storing information, coordinating movement. But somewhere in the middle of all that electrochemical activity, something else happens. There's an experience. There's a what it feels like. When you see the color red, specific wavelengths of light hit your retina, your photoreceptors fire. Signals travel through the optic nerve to the visual cortex, and neurons in area V4 respond selectively to that wavelength. We can trace every step of that chain. But none of that explains why red looks like anything to you, why there's a subjective quality to seeing it, why it isn't just information processing happening in the dark, with no one home. The philosopher David Chalmers, in 1995, gave this a name that stuck. He called it the hard problem of consciousness. And the reason that label resonated so deeply is that it drew a sharp line between two kinds of questions. The easy problems, and he was being a little ironic calling them easy because they're still enormously difficult, are things like explaining how the brain integrates information, how it directs attention, how it distinguishes between wakefulness and sleep. Those are problems about function and mechanism, and even though we haven't fully solved them, we at least know what kind of answer we're looking for. The hard problem is different. The hard problem is explaining why any of that processing is accompanied by subjective experience at all, why there's something it's like to be you. Now, not everyone agrees that the hard problem is actually a real problem. Some neuroscientists and philosophers think it's a kind of illusion, that once you fully explain all the functional mechanisms, consciousness will just fall out naturally and the mystery will dissolve. Daniel Dennett was probably the most famous proponent of this view. He argued, provocatively and at great length, that our intuition that there's some extra ingredient beyond the mechanism is itself a trick the brain plays on us. But plenty of other serious thinkers found that deeply unsatisfying. And the debate is very much alive today. But let's step back from philosophy for a moment, because the real story here is about the scientists who decided they weren't going to wait for the philosophers to settle things. They were going to go looking for consciousness in the brain with the tools they had. One of the most important figures in this story is Francis Crick. Yes, that Francis Crick, the same one who, with James Watson, figured out the double helix structure of DNA in 1953. By the late 1970s and into the 1980s, Crick had grown restless with molecular biology. He'd spent decades in it, won the Nobel Prize, and felt the field had matured to the point where the remaining questions, while important, weren't the kind that kept him up at night. He wanted the biggest unsolved problem he could find, and he decided it was consciousness. So he moved to the Salk Institute in La Jolla, California, and he teamed up with a young neuroscientist named Christoph Koch, who was at Caltech. Together, starting around 1990, they did something that changed the culture of neuroscience. They made consciousness a respectable scientific topic. That might sound strange, but you have to understand the climate at the time. For most of the 20th century, consciousness was considered almost taboo in serious neuroscience. It was too vague, too philosophical, too close to the kind of questions that couldn't be answered with experiments. Behaviorists had spent decades insisting that science should only study observable behavior, not internal mental states. And even after the cognitive revolution of the 1950s and 60s brought the mind back into psychology, consciousness itself, the raw subjective experience part, was still treated as something you didn't really talk about in grant proposals. Crick's prestige helped change that. When a Nobel laureate says this is worth studying, people listen. What Crick and Koch proposed was beautifully pragmatic. They said, look, we're not going to solve the hard problem right out of the gate. Instead, let's look for what they called the neural correlates of consciousness, the NCC. The idea was straightforward. Find the specific patterns of brain activity that correspond to conscious experience, not explain why they produce experience, not yet, but just identify which neural events are present when you're conscious of something and absent when you're not. It was a way of making an impossibly big question into a series of smaller, testable ones. And they had a clever way to do it. They focused on visual awareness, partly because the visual system was already the best understood part of the cortex, and partly because vision offers some wonderful natural experiments. Think about binocular rivalry. When you show one image to someone's left eye and a completely different image to their right eye, the brain doesn't blend them together. Instead, your perception flips back and forth. For a few seconds, you see the image from the left eye, then it fades, and you see the one from the right, then it switches back. The physical input stays constant the whole time. Both images are always there, but your conscious experience changes. So if you can find which neurons change their firing when perception flips, you've found neurons that correlate with consciousness rather than just with sensory input. That's a powerful experimental lever. Researchers like Nikos Logothetis, working with monkeys in the 1990s, did exactly this kind of experiment. He trained monkeys to report which image they were currently seeing during binocular rivalry, and he recorded from neurons throughout the visual system. What he found was fascinating. In the early visual areas, like V1, most neurons just faithfully represented whatever was coming in from the eye, regardless of what the monkey was actually perceiving. But higher up in the visual hierarchy, in areas like the inferotemporal cortex, neurons tracked perception. They fired when the monkey saw their preferred image and went quiet when it didn't, even though the retinal input hadn't changed. This suggested that consciousness wasn't happening at the first stage of processing. It was emerging further along, in areas that dealt with more integrated, meaningful representations of the world. Here's the thing, though. Finding correlates is not the same as finding causes. Just because a neuron fires when you're conscious of something doesn't mean that neuron is generating the consciousness. It might just be along for the ride. Distinguishing correlation from causation in consciousness research is fiendishly difficult, and it's a challenge that the field is still wrestling with. But while Crick and Koch were hunting for neural correlates, other researchers were approaching consciousness from a completely different angle, by studying what happens when it's absent. And this is where things get medically relevant in ways that might surprise you. Consider anesthesia. Every day, tens of thousands of people around the world are put under general anesthesia for surgery. They lose consciousness completely. No awareness, no experience, no memory of the procedure. And here's what's a little unsettling. For a long time, we didn't really understand how anesthetics worked. We knew which drugs to use. We knew the dosages. Anesthesiologists were extraordinarily skilled at keeping people safely unconscious. But the mechanism, what exactly these drugs were doing to the brain to switch off consciousness, was surprisingly murky. Different anesthetic agents have different molecular targets. Some, like propofol, enhance the activity of GABA, that inhibitory neurotransmitter we talked about earlier, essentially turning up the brain's braking system. Others, like ketamine, block NMDA receptors, the same receptors involved in long-term potentiation and memory formation. But despite their different molecular actions, they all produce the same basic outcome: loss of consciousness. So the question became, what's the common thread? What do all these drugs disrupt that makes awareness disappear? One influential answer came from Giulio Tononi, an Italian neuroscientist working at the University of Wisconsin-Madison. Tononi had been a student of Gerald Edelman, another Nobel laureate who had his own grand theory of consciousness. But Tononi went his own way, and in the early 2000s, he developed something called Integrated Information Theory, or IIT. The core idea is elegant. Tononi proposed that consciousness corresponds to a system's capacity to integrate information, not just process it, not just store it, but integrate it, meaning the system generates information as a unified whole that is greater than the sum of its parts. Think of it this way. Your brain has billions of neurons, and at any given moment, it's in one particular state out of an astronomically large number of possible states. The fact that it's in this state and not any of the others is itself information. And crucially, the state of your whole brain can't be reduced to the states of its individual parts, because the parts are so deeply interconnected that they constrain each other. That irreducibility, that integration, is what Tononi thinks consciousness is. He even proposed a mathematical quantity to measure it. He called it phi, which represents how much integrated information a system generates. The higher the phi, the more conscious the system. It's a bold theory, and it makes some striking predictions. It suggests that consciousness isn't limited to brains. Any system with sufficiently high integrated information would be conscious, whether it's made of neurons or silicon or something else entirely. It also predicts that certain brain structures should be more important for consciousness than others. The cerebellum, for instance, has more neurons than the cerebral cortex, roughly 80 billion compared to the cortex's 16 billion or so. But the cerebellum's architecture is highly modular and repetitive. Its circuits process information in parallel, but don't integrate it the way the cortex does. And indeed, you can suffer massive cerebellar damage without losing consciousness. You'll have trouble with coordination and balance, but you'll still be aware. The cortex, with its dense web of recurrent connections, neurons talking to each other in loops, feeding information back and forth, seems to be where consciousness lives. IIT says that's because the cortex has high phi. Now, not everyone buys integrated information theory. Some critics argue that phi is practically impossible to calculate for any real system as complex as a brain, which makes the theory hard to test. Others worry about its implications. If any system with high integration is conscious, does that mean the internet is conscious? A thermostat? The theory has answers to some of these objections, but the debate is ongoing and sometimes heated. There's a competing framework that takes a very different approach. It's called global workspace theory, and it was originally proposed by the cognitive scientist Bernard Bars in the late 1980s, then elaborated with neural specifics by Stanislas Dehaene and Jean-Pierre Changeux in France. The metaphor at its heart is a theater. Imagine a stage with a spotlight. At any given moment, many things are happening backstage. Unconscious processing, sensory signals being analyzed, memories being activated. But only a small amount of information makes it into the spotlight on stage. That spotlit information is what you're conscious of. And the key mechanism is broadcasting. When information wins the competition for the spotlight, it gets broadcast widely across the brain, becoming available to many different systems simultaneously, for language, for decision-making, for memory encoding, for motor planning. Dehaene and his colleagues proposed that this broadcasting is carried out by a network of neurons with long-range connections, particularly in the prefrontal and parietal cortex, that he calls the global neuronal workspace. When sensory information is strong enough or relevant enough, it ignites this network, creating a sudden burst of widespread, synchronized activity. That ignition, they argue, is the neural signature of conscious awareness, and they've gathered some compelling evidence. In experiments where a visual stimulus is flashed so briefly that subjects sometimes see it and sometimes don't, right at the threshold of awareness, brain imaging shows a dramatic difference. When subjects report seeing the stimulus, there's a late wave of activity, around 300 milliseconds after the flash, that sweeps across frontal and parietal regions. When they don't see it, early visual areas still respond, but that late, widespread ignition never happens. It's as if the information knocks on the door of consciousness but doesn't get let in. So here we are, with two major theories, both taken seriously, both with evidence in their favor, and both making different predictions about what consciousness fundamentally is. IIT says it's about the intrinsic structure of a system, how much integrated information it generates. Global workspace theory says it's about function, about information being broadcast widely enough to become available for flexible use. And there are other theories too. Higher order theories suggest you're only conscious of a mental state when you have a higher order representation of that state, when your brain essentially represents its own representations. Predictive processing frameworks propose that consciousness arises from the brain's constant effort to predict its own sensory inputs and minimize prediction errors. What's remarkable is that in the last few years, the field has started doing something almost unprecedented in science, directly testing competing theories of consciousness against each other. A large-scale collaborative project called the Adversarial Collaboration, funded partly by the Templeton World Charity Foundation, has been pitting IIT against global workspace theory in carefully designed experiments where the two theories make different predictions. The first results, published around 2023, were intriguing but inconclusive. They seemed to partially support and partially challenge both theories, which, honestly, is exactly what you'd expect at this stage. We're still early in the science of consciousness, even though it feels like we've been wondering about it forever. And there's another dimension to all of this that connects back to something deeply practical, something that affects real patients in hospitals right now. Because the question of what consciousness is becomes urgently important when you're trying to figure out whether someone still has it. Patients in vegetative states, patients emerging from comas, their families and their doctors need to know whether there's someone still in there, whether behind those closed eyes or behind that empty stare, there's a person experiencing anything at all. And for decades, medicine had almost no way to answer that question. Think about what that means for a moment. Someone you love has a severe brain injury. They're lying in a hospital bed. Their eyes might open and close. They might grimace or move a limb or seem to track something across the room. But are they aware? Are they suffering? Are they perceiving you sitting next to them, holding their hand? Or is it all reflex, automatic brainstem activity with nobody home? The stakes of getting this wrong are enormous, in both directions. If you assume someone is conscious when they're not, families endure prolonged grief, hoping for a recovery that may never come. If you assume someone is unconscious when they actually have some flicker of awareness trapped inside a body that can't respond, the consequences are almost too terrible to think about. The clinical categories here have evolved over time, and they matter. There's coma, where a patient shows no wakefulness and no awareness, eyes closed, no sleep-wake cycle, no purposeful responses. Most comas resolve within a few weeks, one way or another. The patient either recovers some level of consciousness, transitions to brain death, or enters what used to be called a persistent vegetative state. That term, vegetative state, was coined by Brian Jennett and Fred Plum in 1972, and it describes patients who have regained a sleep-wake cycle. Their eyes open and close. They have periods that look like wakefulness, but show no reliable signs of awareness, no purposeful behavior, no evidence that they're perceiving their environment or experiencing anything. The brainstem is working. The cortex, apparently, is not, or at least not in any way that produces detectable consciousness. And then there's the minimally conscious state, a category that wasn't formally defined until 2002, which describes patients who show inconsistent but reproducible signs of awareness. Maybe they occasionally follow a command. Maybe they reach for an object. Maybe they smile or cry in response to something emotionally relevant. It's intermittent and hard to pin down, but it's there. The distinction between vegetative and minimally conscious matters enormously. For prognosis, for treatment decisions, for end-of-life conversations. And here's the thing that should unsettle you. Studies have suggested that the misdiagnosis rate for vegetative state is staggeringly high. Some estimates put it around 40%. 40% of patients diagnosed as vegetative may actually have some level of awareness that bedside examination simply can't detect. This is where the science of consciousness crashes into clinical reality. And it's where a neuroscientist named Adrian Owen changed the conversation. Owen was working at the University of Cambridge in the mid-2000s when he and his team did something that made headlines around the world. They put a patient diagnosed as being in a vegetative state into an fMRI scanner. That's functional magnetic resonance imaging, which measures brain activity by tracking blood flow. And they asked her to imagine doing specific things. They said, "Imagine playing tennis." And when they looked at her brain, the supplementary motor area lit up, the exact same region that activates in healthy volunteers when they imagine playing tennis. Then, they asked her to imagine walking through the rooms of her house, and the parahippocampal gyrus activated, the spatial navigation region, again matching healthy controls perfectly. This was published in Science in 2006, and it was a bombshell. Here was a patient who, by every clinical measure, showed no signs of awareness, no purposeful movement, no communication, nothing. And yet her brain was responding to commands in a way that was indistinguishable from a conscious person's brain. Now, you have to be careful here. Does motor imagery in a brain scanner definitively prove consciousness? That's debated. Some researchers argue that certain automatic processes could theoretically produce similar patterns without genuine awareness. But the specificity of the responses, the fact that different instructions produce different, appropriate activation patterns, made it very hard to explain away as reflex. Owen's patient seemed to be hearing, understanding, and deliberately imagining what she was asked to imagine. She seemed to be in there. Owen and his team pushed further. In a study published in the New England Journal of Medicine in 2010, they tested 54 patients with disorders of consciousness. 5 of them, 5 out of 54, could reliably modulate their brain activity on command. And one patient, a man who had been in a vegetative state for years, was able to use this technique to answer yes or no questions. Think tennis for yes. Think about navigating your house for no. They asked him questions about his life, things only he would know, like his father's name. And he answered correctly. Through nothing but the pattern of blood flow in his brain, detected by a scanner, a man who had been written off as unconscious was communicating. Let that sink in for a second. years in a vegetative state, no outward sign of awareness, and he was answering questions. This work opened up an entirely new field sometimes called cognitive motor dissociation. Patients whose brains show clear signs of cognitive processing, even though their bodies produce no behavioral output. The machinery of consciousness is running, but the output channels are severed. It's like a person locked inside a room with no doors and no windows, fully awake, fully aware, but with absolutely no way to signal to the outside world. Except now, with brain imaging, we've found a way to slip a note under the wall. And this connects back to those theories of consciousness we were talking about, because the theories actually make different predictions about what should be happening in these patients' brains. If global workspace theory is right, then consciousness requires widespread cortical broadcasting. You'd expect to see that signature of prefrontal parietal ignition, that late wave of activity around 300 milliseconds or so. If IIT is right, you'd look for high levels of integrated information in posterior cortical regions. Researchers have actually been developing tools to try to measure something like this at the bedside. One of the most promising is a technique developed by Marcello Massimini and his colleagues at the University of Milan. Starting around 2013, they refined a method called the Perturbational Complexity Index, or PCI. Here's how it works. You zap the brain with a pulse of transcranial magnetic stimulation, basically a brief magnetic pulse delivered through the skull that activates a patch of cortex. Then you record the brain's electrical response using EEG. In a conscious, healthy brain, that pulse triggers a complex, differentiated pattern of activity that spreads and reverberates across cortical areas. It's integrated. The whole brain responds, but it's also differentiated. The response is complex and information-rich, not just a simple wave. In deep sleep, or under general anesthesia, or in a truly vegetative brain, the response is very different. Either the pulse produces a big, simple wave that doesn't spread, lots of neurons firing in sync but without complexity, or it fizzles out locally and doesn't propagate at all. Either way, the perturbational complexity index is low. What Massimini's team showed is that PCI can reliably distinguish between conscious and unconscious states. Healthy awake people score high. People in dreamless sleep score low. People under anesthesia score low. And crucially, people in a minimally conscious state tend to score significantly higher than people in a vegetative state. It's not perfect. Nothing in neuroscience is ever that clean, but it's a measurable, quantifiable index that correlates with the presence or absence of consciousness, and it can be done at the bedside without requiring the patient to perform any task. You don't need to ask them to imagine tennis. You just pulse their brain and listen to the echo. And here's what's really elegant about it from a theoretical perspective. PCI essentially measures the balance of integration and differentiation in cortical activity, which is precisely what IIT says consciousness is about. Hi-Fi means lots of integrated information. PCI is, in a rough sense, trying to get at something like phi without having to compute it directly, which would be mathematically intractable for a system as complex as the brain. So there's a deep theoretical link between the measure and the theory, and the fact that it works clinically is, at minimum, consistent with IIT's core claims. But global workspace advocates would point out that the widespread, reverberating cortical response that PCI detects could also be interpreted as evidence of global broadcasting. When you zap the brain and the signal propagates widely and complexly, maybe what you're seeing is the global workspace in action, information being made available across distributed networks. So even this clinical tool doesn't cleanly settle the theoretical debate. Both sides can claim it. What it does settle, or at least dramatically improve, is the clinical question. And that matters, because families sitting in hospital waiting rooms deserve better than a 40% misdiagnosis rate. Because somewhere, right now, there may be patients who are aware, who are experiencing their environment, who might even be suffering, and who have no way to tell anyone. The science of consciousness, which can seem so abstract and philosophical when you're debating qualia and zombies and the hard problem, that science has real, immediate, life and death applications. And it extends beyond disorders of consciousness into territory that touches almost everyone's life in subtler ways. Because once you start understanding how the brain generates awareness, how it constructs the feeling of being a self moving through the world, you start to understand what happens when those processes go slightly wrong. Not catastrophically wrong, like in a coma, but subtly wrong, in ways that millions of people experience every day. Depression, anxiety, schizophrenia, addiction, neurodegenerative diseases like Alzheimer's and Parkinson's. These aren't just chemical imbalances. That phrase, chemical imbalance, has been repeated so often it's almost lost its meaning. And honestly, it was always an oversimplification. What these conditions really involve are disruptions to the brain's intricate networks, to the way information flows and is integrated across regions, to the delicate balance of excitation and inhibition that keeps neural circuits functioning in that sweet spot between chaos and rigidity. Take depression, for instance. For decades, the dominant story was simple. Depression is caused by low serotonin. The evidence for this came largely from the fact that drugs like Prozac, which increase serotonin levels in the synapse by blocking its reuptake, seem to help some people feel better. But here's the puzzle that nagged at researchers for years. Prozac increases serotonin levels within hours of taking it, but the therapeutic effects don't kick in for weeks. If depression were simply low serotonin, you'd expect the fix to be almost immediate. The delay suggested something more complex was happening. And indeed, more recent research points toward changes in synaptic plasticity, in neurogenesis, the birth of new neurons, particularly in the hippocampus, and in the remodeling of neural circuits over time. The serotonin boost may be just the first domino in a longer cascade that ultimately reshapes how brain networks process emotional information. Remember Hebb's rule, the idea that neurons that fire together wire together. Antidepressants may work, at least in part, by creating conditions that allow healthier patterns of neural connectivity to emerge, replacing the rigid, repetitive loops of negative thought with more flexible, adaptive responses. It's a bit like physical therapy for the brain. The medication doesn't fix the injury directly. It opens a window of plasticity during which the circuits can be retrained. And that retraining takes time, which is why you don't feel better on day one. This idea got a massive boost in 2019 when the FDA approved esketamine, a nasal spray derived from ketamine, for treatment-resistant depression. Now, ketamine is a fascinating molecule. It's been used as an anesthetic since the 1960s. It's on the World Health Organization's list of essential medicines, and it works through a completely different mechanism than traditional antidepressants. Instead of targeting serotonin, ketamine blocks NMDA receptors, those same glutamate receptors we talked about way back when we were exploring long-term potentiation and synaptic strengthening. And here's what's remarkable. Ketamine can produce antidepressant effects within hours, sometimes even faster. People who have been severely depressed for months, who haven't responded to anything else, can experience significant relief within a single day of a ketamine infusion. That rapid timeline tells us something profound. The downstream effects of ketamine include a burst of new synaptic connections, particularly in the prefrontal cortex. Researchers like Ron Duman at Yale, before his passing in 2020, showed in animal models that ketamine triggers a cascade involving brain-derived neurotrophic factor, BDNF, and a signaling pathway called mTOR, which essentially tells neurons to start building new synapses. It's like the brain gets a rapid construction order. And the prefrontal cortex, remember, is the region most associated with executive function, emotional regulation, and the kind of top-down control that depression seems to erode. So the picture that's forming isn't about one neurotransmitter being too high or too low. It's about circuits. It's about connectivity. It's about whether the brain has the flexibility to adapt, to learn new patterns, to break out of the grooves that pathological states carve into neural networks. Depression, in this more modern view, looks less like a chemical shortage and more like a connectivity disorder. Certain networks become hyperactive, others become disengaged, and the whole system loses its ability to shift fluidly between states. And that framing, connectivity, flexibility, network dynamics, turns out to be useful for thinking about a lot of other conditions too. Schizophrenia, for instance. This is a condition that has mystified and frightened people for centuries, long before it had a name. Eugen Bleuler coined the term in 1908, from the Greek for split mind. Though he didn't mean split personality, he meant a splitting of mental functions, a disconnection between thought, emotion, and behavior. The symptoms are often divided into positive symptoms, which are things added to experience that shouldn't be there. Hallucinations, delusions, disorganized thinking. And negative symptoms, things taken away. Flat affect, social withdrawal, loss of motivation. For decades, the dominant theory was the dopamine hypothesis. Too much dopamine activity in certain brain pathways. This came again from pharmacology. Drugs that block dopamine D2 receptors, the antipsychotics, reduce positive symptoms like hallucinations. And drugs that increase dopamine, like amphetamines, can produce psychosis-like states in healthy people. So the logic seemed solid, but just like the serotonin story in depression, the dopamine story in schizophrenia turned out to be incomplete. Antipsychotics block dopamine receptors within hours, but take days to weeks to fully reduce symptoms. They're much better at treating positive symptoms than negative ones. And neuroimaging studies have revealed something more nuanced. It's not that there's simply too much dopamine everywhere. There seems to be too much dopamine signaling in the mesolimbic pathway, which connects the midbrain to the limbic system and is involved in salience and reward, and too little dopamine in the prefrontal cortex, which would help explain the cognitive and motivational deficits. So even within a single neurotransmitter system, the picture is one of imbalance across networks, not a simple excess or deficit. More recent work has pointed toward disruptions in NMDA receptor function as a possible upstream cause, which, interestingly, connects back to glutamate and to the same receptor system that ketamine acts on, though in a very different way. The drug PCP, phencyclidine, which blocks NMDA receptors, produces a state that mimics schizophrenia more completely than amphetamines do, including both positive and negative symptoms. This has led some researchers to propose that schizophrenia may fundamentally involve a breakdown in the brain's predictive processing, its ability to generate accurate models of the world and update them based on incoming sensory information. When that system goes wrong, the brain might start assigning significance to random noise, hearing meaning in meaningless sounds, perceiving patterns and intentions where none exist. Hallucinations and delusions in this framework aren't random malfunctions. They're the brain's prediction machinery running unchecked, generating experiences that feel absolutely real, because the same neural systems that produce normal perception are producing them. Think about that for a second. When you hear a voice, whether it's your friend talking to you or an auditory hallucination, the auditory cortex is active. The experience is generated by the same hardware. The difference is whether that activity is being driven by actual sound waves hitting your eardrums or by internal signals that the brain's error correction systems have failed to flag as self-generated. It's a failure of source monitoring, a breakdown in the brain's ability to distinguish its own predictions from external reality. And that's terrifying in its implications, because it means the line between perception and hallucination is thinner than most of us would like to believe. Your brain is hallucinating your conscious reality all the time. It's constructing a model, a best guess, based on sensory data and prior expectations. Normally, the sensory data keeps the model honest. But when that tethering weakens, the model drifts. Now, I want to shift gears slightly, because there's one more domain where modern neuroscience is reshaping how we think about the brain. And it's one that affects nearly everyone eventually. Neurodegeneration, Alzheimer's disease, Parkinson's disease, ALS, Huntington's. These are conditions where neurons progressively die, and with them, the functions they support. And the story of Alzheimer's research in particular is a cautionary tale about how a field can get locked into a single hypothesis for decades, pouring billions of dollars into it, and still come up mostly empty-handed. Alois Alzheimer first described the disease in 1906 after examining the brain of a woman named Augusta Dieter, who had died after years of progressive memory loss, confusion, and personality changes. Under the microscope, Alzheimer saw two things that would define the field for over a century: plaques, clumps of a protein called amyloid beta accumulating between neurons, and tangles, twisted fibers of a protein called tau inside neurons. The amyloid cascade hypothesis, formalized in the early 1990s by John Hardy and David Alsop, proposed that the accumulation of amyloid beta was the primary cause of the disease, and that everything else, the tau tangles, the inflammation, the neuronal death, was downstream. Fix the amyloid problem, the thinking went, and you fix Alzheimer's. It was a clean, elegant hypothesis, and the pharmaceutical industry bet enormous sums on it. Trial after trial targeted amyloid, drugs to prevent its production, antibodies to clear it from the brain, enzymes to break it down, and trial after trial failed. Patients' plaques were reduced, sometimes dramatically, but they didn't get better. Some got worse. The disconnect was agonizing for researchers and devastating for patients and families waiting for a breakthrough. By the 2010s, the failure rate for Alzheimer's drugs in clinical trials was above 99%. Now, in 2023, there was a partial break in the clouds. Lecanemab, an antibody that targets a specific form of amyloid, showed a modest but statistically significant slowing of cognitive decline in a large trial. It was the first drug to show a clear effect on both amyloid and clinical outcomes. But modest is the key word. The slowing was about 27% over 18 months, and the drug came with serious side effects, including brain swelling and microbleeds. It was a proof of concept more than a cure, a sign that amyloid does play some role, but probably not the singular driving role that the cascade hypothesis originally proposed. The field is now broadening. Researchers are looking more seriously at neuroinflammation, the role of the brain's immune cells, the microglia, which can become chronically activated and start damaging the very neurons they're supposed to protect. They're investigating vascular contributions because the brain's blood supply is critical and often compromised in aging. They're studying the role of tau more carefully, since tau pathology actually correlates better with cognitive decline than amyloid does. And they're exploring the connections between Alzheimer's and metabolic health. There's growing evidence that insulin resistance in the brain may contribute to neurodegeneration, leading some researchers to informally call Alzheimer's type 3 diabetes. The picture, as always, is more complicated than anyone initially hoped. And Parkinson's disease tells a parallel story in some ways. James Parkinson described the shaking palsy in 1817, but it wasn't until the 1960s that Ole Hornikiewicz discovered that Parkinson's patients had dramatically reduced dopamine levels in a brain region called the substantia nigra, a small structure in the midbrain whose neurons project to the basal ganglia and are essential for initiating and controlling movement. The treatment that emerged, levodopa, a precursor to dopamine that can cross the blood-brain barrier, was revolutionary. It gave patients back their ability to move, at least for a time. But levodopa doesn't stop the underlying neurodegeneration. Over years, its effectiveness wanes, side effects accumulate, and the disease progresses. The search for neuroprotective therapies, treatments that actually slow or halt the death of dopamine neurons, has been largely frustrating, much like in Alzheimer's. But here's where things get genuinely exciting, and it's a fitting place to start winding down, because it brings together so many of the threads we've been following tonight. The tools that modern neuroscience has developed: optogenetics, advanced neuroimaging, single-cell RNA sequencing, CRISPR gene editing, brain-computer interfaces, computational models of neural networks. These aren't just incremental improvements. They represent a qualitative shift in what's possible. We can now record from thousands of individual neurons simultaneously in a behaving animal. We can map the complete connectome, every single synaptic connection, of small organisms, and we're making progress on larger brains. The Allen Brain Atlas, a project that began in 2003 at the Allen Institute in Seattle, has created extraordinarily detailed maps of gene expression across the entire mouse brain and significant portions of the human brain, giving researchers a molecular-level atlas of brain architecture that would have seemed like science fiction a generation ago. Brain-computer interfaces, BCIs, are moving from laboratory curiosities to clinical realities. In 2021, a team at Stanford, led by Krishna Shenoy and Jamie Henderson, demonstrated a system where a paralyzed man could type 90 characters per minute just by imagining handwriting, with a neural implant in his motor cortex, translating his intended movements into text on a screen. 90 characters per minute. That's roughly the speed of typical smartphone typing. And in 2023, Neuralink and other companies began human trials of implantable devices, while non-invasive approaches using EEG and other techniques continue to improve. For people with locked-in syndrome, with severe paralysis, with conditions that trap a conscious mind inside an unresponsive body. Remember Adrian Owen's patients. These technologies represent something close to liberation. And yet, for all these advances, the deepest questions remain open. We still don't have a complete understanding of how consciousness arises from neural activity. We still can't fully explain why you experience anything at all, why there is something it is like to be you, lying here, listening to these words, feeling the weight of your body against the bed, aware of the gentle rhythm of your own breathing. The hard problem, as David Chalmers framed it, hasn't been solved. It may not be solvable in the way we typically solve scientific problems, or it may yield to some insight we haven't had yet, some framework that makes the relationship between brain and experience as clear as the relationship between DNA and heredity became after Watson and Crick. What we do know, and this is worth sitting with, is extraordinary. We know that you are, right now, a few pounds of tissue generating an entire universe of experience. Every sound you hear, every thought drifting through your mind, every flicker of memory or emotion, all of it is being constructed by networks of neurons that evolved over hundreds of millions of years, refined by natural selection, shaped by your particular genes and your particular life. The brain you're using to understand these words about brains is itself the most complex object we've ever encountered in the universe. There's something beautifully recursive about that, and maybe the most comforting thing neuroscience teaches us is that the brain is not a fixed machine. It changes, it adapts, it rewires itself in response to experience, to learning, to injury, to love, to loss. Every conversation you have, every new thing you learn, every night of sleep, and sleep, as we touched on, is when so much of that consolidation and maintenance happens. Your brain is quietly reshaping itself, strengthening some connections, pruning others, integrating the day's experiences into the vast, associative web of everything you've ever known and felt and been. So as you lie here now, and your thoughts begin to soften and blur at the edges the way thoughts do when sleep is close, know that your brain is already beginning its nighttime work. Slow oscillations are starting to ripple through your cortex. Your hippocampus is getting ready to replay the day's events, to send them out to the cortical networks where they'll be woven into long-term memory. Your glymphatic system is preparing to flush away the metabolic waste that accumulated during your waking hours. You don't have to do anything. You don't have to direct any of it. This is what your brain does. It has been doing it every night of your life, and it is astonishingly good at it. The 100 billion neurons, the 100 trillion synapses, the electrochemical signals racing along axons and leaping across synaptic clefts, the delicate dance of excitation and inhibition, the vast networks flickering with activity even now, all of it working together to produce this singular, unrepeatable moment of being you, here, tonight, drifting gently towards sleep. And that, honestly, is the most remarkable thing any of us will ever encounter. Not out there in the cosmos, not in the deepest ocean trench or on the surface of some distant moon, but right here, behind your closed eyes, in the soft electrical hum of a living human brain, doing what it has always done, making sense of the world, making sense of itself, and then, when the day is done, letting go and resting